Hey there, I'm Michael Pointer, and this is Abstract Bioscience, a podcast about new and exciting research in biological science taking place across the Norwich Research Park and beyond. Today, we're getting into a rapidly growing field of biology, the study of the microbiome and its effects on and implications for human health and medicine. Each of us carries around with us, in and on our bodies, tens of trillions of bacteria. The bacteria actually outnumber our own cells. These bacteria, which we can call our microbiome or microbiota, are diverse and form complex communities, interacting with each other and with us in ways that we're only just beginning to understand. The majority of our bacteria are found in our gut, but the effects that they have reach far beyond the digestive system. Evidence is mounting that the microbiome has a key role to play in many aspects of human health, from digestion and nutrition through to aging and complex diseases. Among these diseases, cancer is beginning to be heavily studied, with some compelling results. Now, I'm a behavioural ecologist, and I'm much more comfortable with whole animals than I am with cells, so I'm going to leave my intro here and pass over to today's guest, who's Alastair McKee from the Quadram Institute. Alastair, where should we start?
So it's probably easier for me just to maybe define what the microbiota is. I think that's always useful. I think most people now have heard about the microbiome or the microbiota. So first of all, it's important to say that, it's not just your gut that has a microbiota. Almost any part of your body that you think about has got its own microbiota. Our skin has its own microbiota, our mouth, our nose, our eyes. But the gut is particularly in focus at the moment because of its massive influence on how it influences our immune response and also how it helps us process certain things that we eat. So the gut microbiota is made up not just of bacteria, although the bacteria is probably the component that research is focusing most heavily on at the moment, but it's also comprised of fungi, archaea, and a host of other different microbes. And ultimately, there's trillions and trillions of these microbes, whatever they might be. So I think most papers agree that there's likely more microbes in our gut microbiota than there are cells in our body. So we're almost outnumbered by the microbes in our gut. And it's important to understand that they don't act on their own. They act as a community, right? So there's a lot of talk now about how people take probiotics and you're taking one strain of a probiotic and trying to put it into like a massive pool of different things. And that doesn't always translate to a clear beneficial event. Everything acts as a community. So there has to be like a clear balance of the different species that are present in order to get that final positive output.
And what might that output be? What's our microbiome doing?
The two clear things to understand. Firstly, they help break down things like complex carbohydrates. So when people talk about, you need to eat more fiber, our microbiota are really important in breaking down those foodstuffs into nutrients that are usable for our body. So that's number one. They help us produce simple sugars that we can use for energy, etc. But in a more complex way, they also interact either directly or indirectly with our immune system to try and tell our body how we should be responding to diseases. And that can be either positively or negatively, depending on the composition of the bacteria in your gut. So for the purposes of my research, I largely associate the microbiota with bacteria. And so I think that that's the basis of what the microbiota is and why it's important.
What do we know about the effect that our microbiota has on disease?
Most of the research that's been done so far on the microbiota looks at kind of a GI level thing. So when I say GI, it's always the gastrointestinal tract. So things that happen local to the microbiota. And there's been plenty of studies that kind of associate, changes in the microbiota which reduce the healthy members, so the good bacteria, with negative disease outcomes. And where we try and promote a healthy microbiota either through diet or through probiotics, but generally multiple different species, that these result in improved disease outcomes. And those can be cancers or other inflammatory diseases. So the common ones that are known or looked at are inflammatory bowel diseases, so things like Crohn's disease and ulcerative colitis. But in terms of cancer, the one that's had a lot of focus recently is colorectal cancers. I know that recently they've shown, again, in a mouse model, where if you have a depleted level of a particular member of the microbiota. I think the species is called Faecalybaculum rodentium, that it actually promotes an increased rate of tumor growth. It doesn't increase the likelihood of tumors forming, but it increases the rate at which they progress. And in the same study, they showed that by supplementing that species back into your model, you slow the rate at which that progresses. So there's already something there. Without going too into depth in that specific paper, you're showing that promoting a healthy species within your microbiota potentially leads to a positive outcome.
So that's gut diseases and cancer within the GI tract, where the microbiome mostly is situated. But can these bacteria also have an effect on disease in other parts of the body?
In terms of cancers away from the GI tract, there's not a lot known. It's as I say, it's an area that's gaining a lot of traction, and I think in the coming 5 to 10 years, we'll see lots of papers presenting cool and interesting data on hopefully the positive influence that it has. But one of the cancers that I know people have looked at more specifically is melanoma, so skin cancers. There's been several studies shown using different species of bifidobacterium. So bifidobacteria is generally a probiotic species of bacteria that is found naturally in the human gut. By using these bacteria, you can improve the effect of immunocheckpoint inhibitor therapies on melanoma. And actually you see an improved response to shrinking melanoma if you have these bifidobacteria in combination with, it's called anti-PD-1 therapy. So there's not a lot known, but there is definitely studies being done to show that there is this positive relationship.
So people have looked at the effects on disease and on treatments that supplement the good bacteria. But you've looked at consequences of disturbing the microbiota with drugs. So what was the idea behind that?
I guess the reason why we kind of focused on antibiotics is we wanted to kind of show that, yes, you can see good things happening if you use bacteria in this way, but what happens downstream of having used antibiotics in your life, for example, or having a poor microbiota, whether that be antibiotic related or otherwise, how does that affect the downstream, not risk of getting cancer, but risk of cancer becoming worse? And we use breast cancer as the model that we studied.
You worked with mice as a model system to study this stuff. So why is it that mice are used a lot in these kind of studies?
Yeah, great question. So, I mean, I guess the first thing to say is no one wants to use them. It's not the first thing you jump to is using animals in research, but certain questions require a full biological system in order to answer what it is you're looking for. So, for example, we require a gut microbiome that it would occur naturally in the system, i.e. if you feed that microbiota a drug, how it affects that microbiota, and then how that influences the cells at a local level. So, for example, the immune cells in and around the gut, and how that potentially, and what the gut produces in terms of the metabolites that the bacteria produce, like I say, through breaking down things like fiber and other complex carbs, and how that then translates systemically through the blood to the tumor at a distant site. And that tumor obviously has to be in a biologically relevant environment. So we need a breast environment in order to monitor, I guess, a realistic or relevant growth. And so there's no real way of doing that without using an animal model. It's not something that where you can use like organoids or things like that in a lab. You have to have the full system. You have to have all the cell types, all the transportation, nerve pathways, everything. And so that's why we have to use mice to map these effects and understand them in a way that hopefully we can more clearly apply to clinical settings.
So given that background that you talked a bit about, what was the main research question that you were trying to answer with this work?
Yeah, so I mean, I think the first thing to highlight is that throughout our lives at some point, especially into adult life, most people are exposed to antibiotics, whether it's regularly or even once generally to treat bacterial infections, right? But when you're a breast cancer patient or any cancer patient, often you are prescribed antibiotics prophylactically, meaning that if you're going to undergo chemotherapy treatment or you're going to go into surgery, there's potential that your immune system is going to be weakened during that time. And as a result, clinicians will try to help prevent any increased risk of bacterial infection by giving you these antibiotics. And so that was one of the premise that we applied to our model. So essentially, we asked the question, if you have a disturbed microbiota, how does that influence the rate at which breast cancer progresses? So we used orthotopic models, meaning that we induced cancer ourselves. So these are animals which may have got tumors if we let them, but they're not designed to produce tumors. We engraft the tumors ourselves. And so once you have this tumor, how does it grow if you have a disturbed microbiota? And the question we asked was, is it going to grow faster, which is kind of what we assumed would happen with a disturbed microbiota, or potentially would we see the opposite, would it grow slower?
So in order to answer that question, in your experiments, you were comparing the growth of breast tumours in mice that had either been given antibiotics to disrupt their microbiota, or control mice, which instead of an antibiotic had just been given water and so had a normal microbiota. So what were those initial findings?
We started using a very harsh cocktail of antibiotics. So it's not necessarily a mix of antibiotics you would expect to be prescribed by a doctor. But we're kind of looking at, okay, worst case scenario, you have a very severely disturbed microbiota. How does that affect your tumor growth downstream? And we found that it increased quite rapidly versus a control group. So a control group which instead of antibiotics received water. So that was the first thing that we wanted to look at. And one of the questions we initially had was, okay, we see this antibiotics is linked to increased tumor growth, but is it the antibiotics specifically, or is it actually its effect on the microbiota, as we thought it would be? And so we replicated this experiment in germ-free animals, meaning that these animals are born without a microbiota. They've never had any immune education. So when you're a baby, one of the important functions of a young microbiota is to program your immune system to be aware that some bacteria is not necessarily cause for crazy inflammation and crazy immune responses. And so in germ-free animals, they don't have that. So they have no microbiota whatsoever. And we found that by administering antibiotics to germ-free animals, we did not see a change in tumor growth. So that suggests to us and that it is not a direct influence of the antibiotics themselves. It is, in fact, a reduction in biodiversity of the microbiota, or changes in the composition at least, which result in these downstream increases in tumor growth.
Even as somebody who doesn't know very much about cancer biology, I know that there are loads of different types of cancers, not just in terms of where in the body they occur, but how they grow or certain characteristics that they display. So were the tumours that you studied reflective of that diversity?
Yes, they were. So we have access to different cell lines, breast cancer cell lines, and those resemble different molecular subtypes. So they're not exact depictions of a human subtype, but we've got different subtypes which resemble the different types you would see in human beings. So we've tested this not just in one model, we've tested it across at least three. So we've tested it in a luminal A, a luminal B, and a basal-like model of human breast cancer, but these are mouse breast cancers that resemble those subtypes, if that makes sense.
Yeah, I see. And why is it so important that you see this effect across all the cancer subtypes that you tested?
That's actually a super important question to ask. And the reason for that is because obviously in human breast cancer, the different molecular subtypes require different treatment approaches. So for example, in triple negative breast cancer, you wouldn't use hormone receptor therapy because they don't express the hormone receptors necessarily. So you'd have to have a different approach. But with our study, we've shown that across those different subtypes, the microbiota is important every time. So if we deplete it every time, we're seeing an increase in tumor volume, which suggests that regardless of what the treatment is, if in conjunction with your treatment, you try and utilize that influence of the microbiota positively, i.e. by maybe not prescribing antibiotics or only prescribing them when absolutely necessary, you might improve the outcome of the respective treatment for whichever subtype you're treating.
Great. So you saw that giving mice antibiotics makes their tumours grow faster. But as you said, this was with an unrealistically strong mix of antibiotics. How did you try and make this research more applicable to how this might be affecting people?
We started off using a harsh cocktail of antibiotics, which wouldn't be prescribed by a doctor, to significantly perturb the microbiota. So when we did some genomic stuff downstream on this, the cecal and fecal contents, we weren't able to really amplify any bacterial DNA whatsoever. So this is a very significant bacterial knockdown. Then we did inject cells to form this microtumor, and then we leave it to grow and continue antibiotic treatments each week. And at an endpoint, which was generally day 15, so 15 days after engrafting the tumor, we'd identify the differences between our groups, so our control and our treatment groups. And it was always generally that the tumors were larger after the treatment. Once we identified this, the next question to ask is, okay, can you make this any more clinically relevant? And we did this by employing sulflaxin, which is an antibiotic which is used in clinic prophylactically, both at the NNN in Norwich, the Norfolk and Norwich University Hospital, and some colleagues of ours in the United States. And we basically performed the same thing. And we had the same results. Again, we're seeing increased tumor growth after the same treatment regimen. And there was actually very little change in terms of final tumor volume. So the tumor volumes were pretty close, even to the harsh cocktail. However, when we use the sulflaxin antibiotic, it doesn't completely ablate the microbiota. So you're still left with quite a significant amount of different species, but the composition is completely changed. The diversity and shift in the amounts of different bacteria has completely changed.
So that's interesting and maybe slightly terrifying that the effect of using this really harsh treatment is totally comparable to using just a single antibiotic that people are routinely being prescribed. What was the next step in your process?
Then between the harsh cocktail and the sulflaxin pipeline, we saw that there was only a handful of bacteria which were reduced in similar amounts between the harsh cocktail and the sulflaxin treatment. And that was important because obviously with the harsh cocktail, we're ablating everything. So we don't actually know what has gone that might lead to this increase. Whereas with sulflaxin, we know that we can only pick up similarities between these two treatments. And so it's potential that these handful of bacteria are likely influencing our tumor growth. And of those, we, through some literature searches, we found one candidate which kind of interested us the most. And this, again, was that bacteria I mentioned earlier, Faecalibacillum rodentium, or FROD as we call it. And we decided to supplement this back into the harsh cocktail treatment regimen. Again, it's kind of easier to work from a perspective of worst case scenario, how does this pan out, right? And when we supplemented F-rodentium into that pipeline where we're giving antibiotics initially for that first week, then we stop antibiotics and we continue supplementation with F-rodentium, we saw a dramatic reduction in tumor volume relative to the antibiotics. It returned back to the same size as the control. So you're rescuing that effect of the harsh cocktail. You're rescuing that inflamed growth kinetics. So we identified that, yes, these good members of the microbiota are important at regulating the rate at which our primary tumors are progressing.
That's a really nice result. I noticed there that you said progressing. And that's one thing that I've been wondering about. Are disease progression and tumor growth the same thing?
Yes and no. So I think in the context of our paper, we tend to use the term disease progression in line with tumor growth, right? Generally speaking, the larger your tumor grows in human patients, the more likely it is going to be that it's at a stage where it might metastasize. That's not exclusively what drives metastasis. There's a whole host of different tumor microenvironment elements which promote metastasis, but generally the larger your tumor, the later stage it's likely to be, right? So we kind of use that in this context. Largely, disease progression would mean moving from a primary stage to a secondary stage. So potentially, yeah, for clarity's sake, we're not saying that these changes definitely promote increased rate of metastasis. So we're not saying you reach that point quicker. What we're saying is that when that happens, the same thing happens. Those tumors grow bigger, faster, right? So the last thing you want is if you do have secondary breast cancer, the tumors in your lungs to be growing quicker, or the tumors in your brain to be growing quicker. And that's the important observation that we make there. Having said that, there has been a study done by colleagues in Virginia a year or so before our paper was published, which showed similarly when they used a harsh cocktail of antibiotics in a different model. Actually, it's a model that we used, but it was a different treatment regimen than what we used, that they saw an increased rate of metastasis. So what we have observed is not specifically the only thing. It's kind of one of those moments in science where you have to kind of piece everything together, take all the puzzle pieces together and try and draw a conclusion from it. So I think there's definitely a role that the microbiota does play in the risk of metastasis, but in our paper, what we are stating from scientific observation rather than our own theory is that we're not seeing an increased rate of metastasis or increased number of metastasis at least. we are seeing an increase in the size of those lesions in the secondary tumor.
Okay, let's have a recap there. So you saw these effects increase tumour growth when mice were treated with either the antibiotic cocktail or just sulflexin, the single antibiotic. And you saw this for primary breast tumours and secondary tumours after the cancer had spread. And you saw that if you supplement a species of good bacteria back into these antibiotic mice, the tumours grow more slowly again. But next, you went looking for the mechanism, what it was that's actually causing the tumours to grow more quickly, right?
So I guess the first thing to say is when we observed this increase in tumour growth in both the sulflexin and the harsh cocktail treatments, we kind of naturally assumed that there was going to be a handful of immune cells which might be responsible for it. So generally in cancers, in most cancers, the immune system plays a major role in progressing those cancers, both in terms of increasing the rate of primary tumour growth as well as metastasis, etc. And they're kind of generally well known. So there's a lot of information on things like the change in behavior of macrophages in a tumor environment and how those can either, generally they have a kind of bad name where they increase the rate at which tumors grow and spread. So we did a whole host of different things looking to see hopefully these candidate immune cells shifting between our things. So maybe seeing an increase in macrophages or increase in certain T lymphocytes and a reduction in anti-cancer T lymphocytes. But we didn't see these, and it was kind of surprising to us. In either sulflexin or the harsh cocktail, we didn't see these. And we were fortunate that we had the opportunity to undertake what is not extremely new technology, but fairly new technology, where you sequence individual cells to look at what's called their transcriptome. So what genes are activated and readily being told, we need to use you, right? So I guess in broad terms, you're looking at possible changes in cell behavior through the single cell RNA sequencing technology. And so we sequenced a couple of tumors from our harsh cocktail and our control and tried to identify what cell types, if at all, what differences in the cell types were there. So it's not the best way to identify cell type, but when you've got a very complex makeup of cells, it is one way that you can try and do it. So different cells have got different protein expression, and you can try and use those to pick apart what that cell might be. But also, once you've identified those populations, you can try and compare between your treatment and your control what genes are more activated in one versus another.
So you're looking at the genes that are switched on in these cells that you've taken from tumours, and by comparing your data to known profiles for different types of cells, you can then say our measurements match with such and such a cell type, so we can be reasonably confident that that's what our cell is. So what cells did you find?
One of the things we identified was in our antibiotic group, the tumours had an increased expression of genes associated with what's called tumor stroma. So this is generally kind of edge of tumours, around the edge on the outside, and it consists of a milieu of different cells, including immune cells, which are often associated with a worse prognosis. So what we decided to do was histologically look at our tumours. We didn't see a clear difference in the amount of stroma, but what we did see is granular looking cells in the stroma from our antibiotic tumour versus our control group. And so again, we hit the books, so to speak, and we try to identify what granular cells might be present in the stroma of a breast tumour. And one of the things that came up was mast cells. So a mast cell is an immune cell which generally is associated with like allergy responses. So they're actually a major player in the gut, and they essentially produce things like histamine and tryptases, which are basically all these immunoinflammatory biomolecules that trigger heavy immune responses and ultimately can result in an allergic reaction and anaphylaxis. But in the context of cancer, that doesn't mean a whole lot to us, right? Because cancer is not an allergic response, so to speak, or at least that's our belief as it is, right? So we were kind of surprised to see that these cells were mast cells, and there was a clear increase in the number of these mast cells, not just in one tumor model, but several. So we identified it with colleagues in Italy, with colleagues in the States, and there was always this consistent increase in the number of mast cells in tumors which had been taken from animals treated with antibiotics with a perturbed microbiota.
It seems then that the mast cells could have something to do with making the tumours grow more quickly, but how did you nail that down?
So what we wanted to identify is, okay, let's firstly confirm that we think these mast cells are influencing tumor growth. So what we used is a drug that's actually used kind of commonly to treat things like eye allergies and an array of other allergy responses, which blocks the activity of mast cells, so it prevents them from generally performing their major function. And we administered what it's called cromolyn. We administered cromolyn to animals that were treated with antibiotics and animals that were treated without antibiotics, so with water. And we compared those to animals which didn't receive cromolyn but did have either water or antibiotics. And all of these were tumor-bearing, we call them. And what we observed was that in the animals that were subject to antibiotic treatment and cromolyn, there was a significant reduction in tumor volume versus the animals with just the antibiotic, right? So it's appearing that this cromolyn is preventing these mast cells from doing what they're doing to promote tumor growth, and we're rescuing that effect. What was interesting, though, is in the control groups where we had control and then we had control and cromolyn, we saw that cromolyn didn't have an effect on tumor volume. So what that suggests to us is when we give antibiotics, the microbiota is becoming disturbed, and that disturbance is somehow triggering a specific behavior in mast cells, which is promoting this increased tumor growth. It's not the result of mast cells specifically, because otherwise, if we gave cromolyn to control group, we'd see this shrink in tumor volume versus control, but we don't. So it is a microbiota-dependent effect on mast cells, which is pushing this tumor growth up.
Fantastic. I really enjoyed the paper. It's been fascinating to hear more about it today, and I'm looking forward to seeing where it goes. It seems very plausible that it could be applied to really helping people in the future.
Yeah. From my perspective, it's exciting because this is probably one of the few papers linking changes in the microbiota to breast cancer in a model which is kind of comprehensive. So because we use an animal model, we can try and look at not just direct effect of antibiotics or direct effect of different things. You can see the whole system working together and how that influences disease progression. So I think the exciting bit for me is that, we're showing that the microbiota has a really positive role in potentially regulating its influence on cancer at a site that's completely away from the GI tract. And I think that's a really important message to show, one, the importance of the microbiota, but two, the possible influences or the possible things we can target to try and reduce the progression of breast cancer using the influence of the microbiota.
That was Alastair McKee from the Quadram Institute. Thanks to Alastair and to his co-authors on the paper, which is titled Antibiotic-Induced Disturbances of the Gut Microbiota Result in Accelerated Breast Tumour Growth. As usual, there's a link to that in the show notes. This is getting to be a longish episode already, so I'm going to wrap it up there, but I hope you found that as interesting as I did. There'll be more abstract bioscience very soon. In the meantime, show notes, transcripts, details about the podcast, social media accounts, and links to information about the Norwich Research Park can be found on the Abstract Bioscience website at abstractbioscience.co.uk. The show is written and presented by me, Michael Pointer, and you can contact me at abstract.bioscience.podcast@gmail.com. We're on Twitter and on Instagram, and our excellent theme music is by Phoebe Troop. Thanks for listening. Catch you next time.